Good morning or good afternoon, everyone, and welcome to today's webinar, depending on where you're joining us from. My name is Tarek O'Dali from Business Review, and I will be your host. It is our pleasure to have Catalent Pharma Solutions with us today. He'll be presenting this webinar titled Expedite Your Early Drug Development with Unique Pre-Formulation Techniques. Today's guest speakers are Michael J. Hagerman, PhD, former Executive Director of Discovery Pharmaceutics at Bristol Myers Squibb. Also, we have Dr. P.Y. Chen, Director, Early Development at Catalent Pharma Solutions. I'd like to welcome you to our webinar platform On24. You'll notice this webinar is browser-based. If you do disconnect for any reason, please click on the link you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. Just type them into the box at the top left-hand corner of your screen and click Submit. We'll try to allocate some time at the end of the session to address any questions or thoughts you may have. There is a survey available today, and it is available throughout the session. Please answer the question in there for feedback if you get the chance. Please use the yellow Help widget if you do require technical assistance. And you can move, resize, or maximize any of the windows in front of you to get a better view of the slides. But now, that having been said, please allow me to welcome today's first speaker. Michael, over to you.
Thank you very much, Tariq, and welcome to everyone. Good morning or good afternoon, as it may be. What I hope to do today is talk a little bit about knowing your molecule and the importance of pre-formulation and setting the stage for that overall process. And the things that we would like to cover is pointing out that not all drugs are created equal, and it's important to recognize that. We'll talk about what that means. A brief discussion of a preliminary target product profile. So it's often difficult to understand why you should be thinking about this so early, but it has a significant impact and interaction with pre-formulation information. Then we'll get into the translation of the actual molecular structure. So this is really knowing your molecule to how that relates to physicochemical properties of that molecule and beyond. And then we'll talk about, I've kind of split it up into three different areas. One is the bioperformance area, which physicochemical properties have a key role in. And this is really the interface between biology and physiology with the physical chemistry. And then we'll talk about manufacturing and processability, and this interface being that of formulation and process engineering with the properties of those molecules, and in particular, more importantly, derived properties of the composite materials. Finally, the shelf life, which involves the interface really with the end user and the marketplace, and has a significant role in the end success of the molecule. Lastly, we'll just mention how important it is to have the integration of this pre-formulation information that gets generated to design formulation strategies and help design processing strategies based on the capabilities that you may be working with. What we find, I think, is we often, and this gets to the old somewhat proverbial haystack and trying to find a needle in the haystack and presuming that all molecules would be similar in property. Actually, as would be noted in the discovery environment, you get molecules of all sorts and types, and actually anywhere from liquids and syrups to very oily molecules to very highly structured bricks sort of molecules, as well as even as we get into larger and larger molecules, more of the polymeric type structures that have specific phys-chem properties that have to be dealt with. So what really happens in this discovery space is very important because it's part of the design phase of the molecule. And a point where you're trying to balance a number of different things, that is both structure-activity relationships and structure-property relationships. Just pointing in this slide, one example, we won't go over this all, but if you look at the upper right, you can see how you have to balance these, and sometimes that depending on the substituent that you put on there, the effect that it will have on activity versus metabolic stability versus solubility can all be in different directions. And so we end up having to do a balance. And this comes across the entire structure of the molecule and is often the reason that sometimes thousands of molecules are made and iterated through to actually end up with a molecule that goes forward. I think what's important in this, while we're going to talk a lot about pre-formulation of a molecule that you know what you have, it is always better to design in developability and progressibility into the molecule than having to rely on risk mitigation in development. Sometimes it simply isn't possible, and we have to be cognizant of that and make sure that the pre-formulation data that gets generated will support further progression. Early on in this process, most of the drug discovery activity actually tends to focus on bioperformance and less so on the developability. And we're not going to go through this number of different formulation sorts of strategies that are used in the discovery environment, but just to indicate that they can be very varied, and actually they end up being highly dependent on the properties of the molecule as well, even though you often have very little information at that point in time. A lot of people ask me what my brick is in the bottom right-hand corner with the handle on it. That's my prodrug handle, and I think that's important to always keep in the back of our mind with regards to synthetic handles that can be used to overcome physicochemical properties. As we go backwards to the molecular properties, many of these molecular properties are often properties which can be computationally determined, and there's a number of software packages which can help with that. The ability to effectively translate those molecular and structural properties into physiochemical properties is very important, and in part is one of the reasons that it becomes more difficult to do computational calculations of the physiochemical properties themselves, such as solubility and permeability, because they end up relying on a number of molecular properties. So you begin to get a multivariable situation with variability and error associated with each of those computations. So what we find is that we tend to measure the physiochemical properties on a much more regular basis. And then those physicochemical properties in turn would have an impact on the solid form properties that we often encounter and try to move forward. So if you look at the top schematic, really we're going from molecular properties to the phys-chem properties to the solid form. And as we go on, then those properties all come into influencing and impacting the formulation and manufacturing properties. That is how we go about filtering material, isolating, sizing material. And I can't emphasize this enough that actually I think we often kind of neglect the processing considerations for isolation of API and the impacts that it has. And clearly our ability to have a reproducible and robust process for API production is highly dependent on these physiochemical properties and understanding them, as is the formulation strategy that evolves out of that. And then that all comes together to actually carry us toward the shelf life properties. And I think the one thing that becomes important is that while we go through this and optimize and make sure that we have chemical stability, physical stability, that we have a product we can manufacture, we have a product that we can package and ultimately market, the key thing we can't walk away from is that you have to maintain bioperformance. This next slide, we don't really have time to go into it, but actually the translation of molecular properties also occurs into the preclinical and clinical attributes. So that is the properties, the biochemical properties such as target binding and tissue partitioning, membrane partitioning, transporter binding, those all are also related to these physiochemical properties and molecular properties, which ultimately then translate into both the ADME properties and the pharmacology and toxicology properties. So again, this gets back to the point of trying to optimize a number of different parallel properties, and so that you can see that this train from molecular properties to pharmacology and toxicology is one that is occurring simultaneously with our desire to move from molecular properties to a marketable product. I just wanted to take just a brief minute to talk about a preliminary target profile and why that's important. And it really comes down to it provides the basis on which developability hinges. For instance, to understand what it is, what your indication is, what the competition is, are we going with an oral product or IV? Obviously, the physiochemical properties become very different. It's also challenging at very early stages of discovery to understand what the dose might be. However, it is extremely important to always keep that in mind as you're going and as it continues to be refined. The transition from the dosage form to drug for absorption. So this really is what dictates the bioperformance, is whatever mechanism or formulation strategy that we choose, whether it be a solubilized drug to overcome dissolution problems or a solid dosage form, which then liberates the granules and ultimately fine particles for dissolution and absorption, are critical. While this process is going on, we're always battling, especially with the poorly soluble drugs, this tendency for the drug to precipitate out, irrespective of how we formulate it. So it's a kinetic phenomena which we have to deal with and be aware of. And so this really gets back as well to the pre-formulation properties. In an effort to have the drug actually be absorbed, it has to be in solution. And again, while we won't go into it today, this really relates to the thermodynamic activity of the drug and the recognition that in many cases when you solubilize a drug, you haven't necessarily provided free drug for absorption. So those things have to be balanced as well. The bioperformance in this interface with both biology and physiology further complicates the situation in that now you see that the fraction absorbed is actually really the interrelationship between permeability, solubility, and dose. And you can see that permeability can vary over 2 to 3 orders of magnitude, while solubility can vary over 5 to 6 in a practical sense, and dose can actually vary 3 to 4. And it becomes important to recognize that both are important in how they interact for optimal absorption. What also becomes important is the dose, and in particular, very early investigations where you're having to do drug safety evaluation. While the dose in drug safety may actually vary over the same 3 to 4 orders of magnitude, what you find is that it is actually at higher doses because you're pushing the system to generate information with regards to both hazard and safety. So it often becomes the case that the formulation strategies at these higher doses needed for toxicology are significantly impacted. And the pre-formulation information becomes extremely critical in just designing the formulation strategy for the tox study. In fact, it's often the case that the formulation for toxicology is as challenging or more challenging than would be the formulation to obtain bioperformance at a typical therapeutic dose. It also, one has to recognize that as you do this, the formulation strategies are such that it's unrealistic that you can always dose the same formulation in a tox study that you can dose in a efficacy study. And to understand how the doses translate and the impact that they have on the results, the pre-formulation package once again becomes very critical. So what are some of the key attributes which are characterized in pre-formulation? And PY is going to go into this a lot more, so I'm just introducing it here. But there's significant physiochemical characterization that occurs, and what you find is very early on the solid form characterization becomes quite important, and understanding the thermal properties of your molecule, certainly the various forms of the molecule that can be present, as well as how they behave in various media over a pH range. As you continue on, the particle size and particle distribution and their impact on dissolution become critical, and so you really need to have an understanding of the dissolution properties as well. This is just a slide which really I want to point out that the solid form characterization, the more you know about it, the sooner, the better off you're going to be. And in particular, it will allow and simplify much of the translation that occurs as you move from one process to the other, ensuring that you're comparing similar drugs. Now, it may be that very early you don't necessarily have the optimal forms present, but it's crucial that you know what form you do have so that you can make comparisons back and understand when you see differences and what sorts of differences forms may make at later stages. Obviously, a key role in these forms as it translates to bioperformance is both solubility and dissolution. And whether that drug particle then is crystalline, co-crystalline, you make it an amorphous dispersion, all of those things can impact the rate of dissolution. And as everyone knows from the Noyes-Whitney equation, the surface area becomes important, so particle size becomes important. As you increase or decrease the particle size and increase surface area, you can increase your rate of dissolution. Similarly, as the solubility, the saturation solubility at the interface goes up, you can increase the rate of dissolution. The other thing that's important to recognize is, in particular, if you have ionizable compounds in that diffusion layer, the reactive gradients that can occur there with regards to pH buffer, counterions, as well as solubilizing agents and the role that they have in carrying molecule away from the solid surface and into the bulk media. So these all play a significant role, and it becomes important to understand that role. What I wanted to do as we finish up here and I transition it over is just provide a few examples of the sort of impacts that we see very early on, and it becomes valuable to get ironed out. And this is one of suspension particle size. You can see that in this particular case, with a free acid, which you can solubilize by taking the pH up in a non-buffered solution and dosing that, you get very good exposure from the solution. You can see that even with a submicron suspension, which is had a D50 of about 0.6 micron, you get limited exposure. And then you can see that if the unmilled suspension, 30 micron, you get almost no exposure. So it's very valuable to understand what the impact of particle size is going to be as you move forward. Similarly, the use of a salt to overcome dissolution rate problems is a very common strategy, especially when you have an ionizable compound, in this case a weak base, in which we've got the hydrochloride salt. And you can see that you can get a huge bump with regards to exposure. And this is very common, and one of the, probably because of the industry's lack of familiarity with salts and their use, is often one of the preferred strategies for going forward. It's also important to understand the ionization properties of the molecule, in particular as it transverses the GI tract. That while you might have the adequate solubility and dissolution properties for the hydrochloride salt, for instance, you may actually get in a situation where as you traverse the intestine, you actually get to a point where the solubility of the weak base at intestinal pH is significantly below the solubility, and so therefore you end up getting precipitation of the drug and reduced exposure. It becomes very important when you interpret even your animal data with the variability that one often sees in dog pH, in the intestinal pH of dogs and the stomach pH, to understand whether the effect of pH and solubility is going to play a role in your data. This next one, and this is very commonly used, certainly in the last 10 years because of the advancements in being able to pursue developability of amorphous systems and amorphous dispersions. There's been much more extensive work done in this area in which you're looking at utilizing an amorphous solid dispersion or actually salts or solubilized formulations as well, in a case where they all, as shown by arrow number 1 here, can bump the drug into solution at a much higher concentration than the saturation solubility, therefore giving enhanced driving force. The next key is how long you can maintain that supersaturation, as indicated by arrow 2, through the use of precipitation inhibitors and potentially solubilizers, and keeping the drug at high concentrations. Then once the drug does precipitate, as occurs through process 3 indicated here, what is the size and form, what's the nature of that precipitate, and how fast does it redissolve? So those things can all play a very significant role in understanding the process and developing strategy. This is just an example of a powdered microdissolution study with both crystalline, solid, and dispersion formulations. You can see at the bottom of the plot the crystalline drug goes into solution and reaches a steady state. By creating a solid dispersion, in this case either with PVP or with HPMCAS, one sees that you can get a much higher, almost tenfold increase in peak solubility, and then you see a very big difference in this particular case between the two different polymers with regards to how long you can maintain that particular enhancement in solubility. And so what you see in this case actually is with the HPMCAS, another factor which comes into play is the nature of the material that precipitates out and the fact that we're ending up at a different equilibrium level of solubility than we are with the crystallines. So we actually have some sort of higher energy form that's likely precipitating out. In many cases, it actually can be a co-precipitate with the polymers that are involved. The impact that this has is shown in this slide, in that you get much more linear response. If you look at the very bottom of the plot in the square symbols, which is really a nanosuspension, so you've reduced the particle sizes to as much as practically possible, and you can see that you get very little exposure. and in some cases even going with a solubilized formulation where you've added TPGS in this case as a precipitation inhibitor, you can get enhanced exposure, but then you see it plateaus off. At some point, as you continue to increase dose, you begin to see pretty much complete precipitation of that. What this really allowed us to do in this particular case, we had a target area under the curve of about 1,500 micromolar hour, and this was to conduct a tox study. And to do that, we were able to accomplish what we needed to by going to the spray-dried dispersion. This does not always necessarily mean that you would have to develop a spray-dried dispersion, but it certainly enabled the toxicology studies. In closing, and before I turn it over to PY, I just want to mention again, this process of creating the strong pre-formulation package is important because we're moving from bioperformance to overall performance of the materials. And regarding developability and progression, being able to make a decision whether the molecule can be salvaged or challenges mitigated through formulation and processing. The data the pre-formulation data will underwrite your in vivo data and provides the basis for your formulation strategy and processing options. Very important is it can often avoid reformulation work due to solid form issues later on. And facilitate regulatory responses and possible approval. So it allows you, when questioned, to respond much more effectively, ultimately giving you a competitive advantage. So I will turn it over now to PY to go ahead.
Thank you, Mike. That's a great introduction to a very broad and interesting topic. For the next part, I'll try to focus on the practical aspect of the drug development. So if you handed a drug candidate molecule, what can you do and how can you do it, get it done quickly? So I'll start with a broad picture at where this actually fit in the broad spectrum of the drug discovery and development. I think most of you are familiar with, as much as we know about biology, chemistry, and physiology, medicines, and how we develop them, and the reality, the attrition rate is still extremely high. For the molecule that comes to preclinic and into the clinic, and the attrition rate is greater than 90%. So that's one. And it takes a long time to get a molecule through the drug discovery and development process, typically around 14. And it varies a lot depending on the type of program that you're working with and the development strategy. And it also costs a lot to get a molecule successfully to the market, well over $1 billion. So with this, where does the pre-formulation, preclinical, on the grand scheme of things, it's a relatively short period of time. As you can see, I kind of highlighted on this slide, it takes about 4 or 5 years to get a preclinical candidate for us to work with. And we have roughly about 1 year to do that work, to prepare and get everything ready, to do the APS synthesis, to do the pre-formulation study, to get the PK safety assessment and formulation, everything ready for clinical study. And on the back end, as you progress, it gets more expensive. So I think the reality is we have to balance the time and resource and get there quickly without compromising the objective, of course. So here are a little bit more challenges in early development. Specifically, we typically work with very limited amount of material at early stage. At late discovery, early development, you're talking about milligram quantities or maybe gram quantity of material for for us to perform various tests. And we talk about aggressive timeline. And on top of that, despite various efforts to get molecules that has good developability profile, there are still a lot of molecules that comes with a lot of technical challenges. Specifically, over 70% of the NCEs comes through has poor solubility or break dust-like property. And for this type of molecule, the typical simple formulation, powder in bottle, powder in capsule, probably will not work, like Mike pointed out. And you need some bio-enhanced formulation to enable the tox and from clinical studies. Again, this is kind of a rehash of what some of the stuff that Mike mentioned. Specifically, you want to get the pH solubility profile of your molecule as well as the stability. And doesn't really take a whole lot of compound to do that, as I'll show you on the next slide. And you can actually calculate the partition or measure the partition coefficient of permeability. So these are things that you can do and assess very quickly with relatively limited amount of material, resource, and time to really set you on the right stage for your development. And specifically, I'm going to focus on the solid state property. As Mike mentioned, there are quite a few of them, the melting point and a number of them. And you can manipulate these properties and modify them to achieve the goal by choosing various options like salt, different polymorphic forms, amorphous forms. There are a lot of options. So the question is, how can you assess your molecule and assess various options and really pick the right option for your development? That's really the challenge and the goal that we're trying to achieve here today. One of the tools that you can use is the biopharmaceuticals classification system for oral drug delivery. Hopefully most of you are familiar with this. It really takes into consideration of three very simple factors: the permeability of your compound, the solubility of your compound in fasted state simulated gastric fluid. That's really like because most drug absorption occurs in small intestine, where they have the largest surface area for absorption to occur. The other element is the dose, the projected dose. If you have these three factors for your molecule, you can actually do a simple calculation put on this map. It basically classifies the molecule into 4 different classes. Class 1 with good solubility, good permeability. That's a very easy and straightforward case. And a lot of molecules actually fall in class 2 or class 4, where you have, class 2 you have good permeability and poor solubility. So, and class 3 you have good solubility but poor permeability, and 4 you have poor solubility and permeability, and that's the most challenging category to develop. The developability classification is a slight modification of that, and it's actually evolved over in GSK. The basic modification for that is really they increased the volume to 500 ml instead of 250 in the BCS system. And the other thing they added is this SLAD line for this class 2 compound. The idea is for a highly permeable compound, you don't need to dissolve the entire dose. As you dissolve partial dose, it gets absorbed and removed from GI tract, and additional material can dissolve. So it divides that category into 2 classes: dissolution rate limited or solubility limited, like 2B in that case. So the idea is to use this to drive the formulation development. On this slide, I have just show a basic pH solubility profile. Again, there are a couple of ideas I want to share here. One, you can get this done fairly quickly with limited amount of material, depending on the solubility of the compound. It can be as little as maybe 100 milligrams of material. When you do this, and if your compound is crystalline, and try to use the crystalline form, as Mike pointed out, amorphous material, different solubility form can have a very different solubility. And if you start with the right form, you can get the data a lot quicker. That's one. The second message that I really want to share is about the salt selection. For this compound, this is actually a free base with a pK about 7.2. And if you measure the pH solubility, and you can actually do a fit and calculate the pKa of the molecule. The other thing is that for salt, this concept of pH max for two different salts, salt 1 and salt 2. Salt 1 has lower solubility, and salt 2 have higher solubility. And I have the two arrows pointing there. It's like pH max. So at pH higher than the pH max, even if you start with a salt, it's going to have a tendency to convert back to the free base form. And in order to take advantage of the salt, you really have to keep the pH of your suspension or solution below the pH max. So that's actually a very important concept to keep in mind. And the other thing is, like, having this, you immediately know, like, what kind of solubility enhancement can I get at doing pH adjustment. So it serves a number of purposes. Here again, like a slide talking about salt selection, roughly like, greater than half of all the small molecule drugs on the market are developed as salt forms for various reasons, like Mike pointed out. The number one reason is probably the solubility that we are all familiar with. And on the right-hand side, there are a number of very common known drug products. It has the solubility to the free form compared to the various salt. And you can see you can have orders of magnitude enhancement just simply by selecting salt. The other reason is the chemical stability. And by adjusting pH, by making different salt, you can actually address the chemical stability of your molecule. And the physical property we talked about a lot, and also this impacts the API isolation and the formulation development strategy. Here's an example. It's actually a very well-known example. It's the Praxair. The active is olanzapine by Eli Lilly, and it's indicated for schizophrenia. So for this particular population, the patient compliance is a key issue. So that's why there are actually multiple dosage forms developed to address that patient compliance. So the compound is also is a BCS2 or DCS2 class. It's highly permeable and poorly soluble. And the free base form was used for tablet formulation. And they actually did a salt screening. They found more than 20 salts that considered developable, but with various, developability profiles in terms of solubility, chemical stability, manufacturability. So what's interesting, there are two things I want to point out. One is they developed a Zydus formulation, which is a patient-centric formulation. It's orally disintegrated tablet you can take without water. So that's that. The other one is a long-lasting, extended release injectable suspension using poorly soluble permoyant salt. And a lot of times we were talking about enhancing the solubility by using the salt. And in this case, we actually take advantage of the poor solubility and slow release to achieve the slow release, long-lasting PK profile. And this one injection can last 3 to 4 weeks. So. Here's an example that I actually personally worked on when I was at GSK, and we published this as a poster in one of the meetings. So this is a molecule. Again, it's like DCS2B. It's a pretty soluble molecule, and hydrochloric salt was selected for development. And this salt has actually many different solid-state forms. We found actually 28 different solid-state forms. These forms basically, there's a form 1, non-solvent anhydrous form. There are multiple hydrates, and there are solvates, multiple solvates. The reason I picked this, it has very complicated polymorphism, and initially we did not even know there was acetonitrile solvate because that was picked as a process solvent. And later on, at a very late stage during phase 2B and on larger scale, the acetonitrile solvate popped up, and that caused a lot of headache to mitigate the risk and really resulted pretty long delay in the project. Here's a phase diagram of, in the process solvent. On the left-hand side, you can see it's the temperature, and x-axis is the percentage of the water. It's like the process solvent is like acetonitrile water mixture, and so you manipulate so this is a phase diagram that shows which solid-state form is stable under process conditions. So I don't really have time to go through the details, but the high-level message and bottom line is if you understand your system, and even though it may be very complicated, you can find a process to make the right form. So. For the next few minutes, I really want to talk about, the specific screening platform that we have been using and how to think about the solid-state form screening selection. So I'll start with this one, looking at the pharmaceutical impact of the solid-state forms. Mike already pointed out all of them. The way I look at this and I talk about this, I think it's this material science tetrahedral on the right-hand side. If you think about the solid-state form, and it has an impact on the API purification and isolation process. It certainly has impact on the drug product manufacturing process, especially for solid-state dosage forms, where the physical property, the bulk of physical property will have significant impact. And of course, we all know it has huge impact on the material property, the solubility, dissolution rate, stability, both chemical stability and physical stability, and the hygroscopicity, bulk density, flow property of the material. So, and most importantly, it has a big impact on the drug product performance, like the bioavailability, efficacy, and shelf life. So the high-level message here, again, is like all these are interconnected, and by choosing the right solid-state form, you can actually simplify the drug development. And it may not be the particular salt or polymorphic form that you choose may not be the best for any one of this, but the goal is really to pick one that's easy to manufacture and that has the right property and the right performance, and really balancing all those factors. On this slide, I've just shown like some practical considerations. For solid-state form screening and selection, there are a lot of different options, like Mike showed you earlier on that slide. So the question is, for your particular molecule, what can you do quickly to assess what options do you have and what are their properties? how are you going to make them, and whether or not they fit your development strategy. So there are a lot of variations. There are a couple of things I want to point out. I mean, this work you can do as little as you can and use a relatively limited amount of material, or you can also do a lot of work and spend a lot of time and material on it. So you really need to choose the right strategy to do the right amount of work for your compound. Typically, most of the salt screening will be done at an earlier stage if your compound has an ionizable functional group to form the salt. And that typically, for most companies or most programs, people try to get that done early so you can use the right salt form for the preclinical safety assessment, tox studies, and PK evaluations. And that also allows the chemist to develop the process to make that form. And typically, once you select the salt, the polymorph screening will be done on that particular salt. And most companies will target to get that done and get the form selected, and the right polymorphic form of the right salt are used for clinical studies. And so with that strategy, you can take advantage of the right properties of this and also avoiding bridging PK or safety studies down the line. For the next couple of slides, I want to show you a couple of the specifics about this Optiform technology, the high throughput platform that we being using to conduct these studies. This platform actually was developed in GSK. It was acquired by Catalent about 7 years ago. And it's very unique in terms of how it's designed and how we actually use it. It's being used, very extensively supporting like GSK portfolio back then, and now the last few years we have been using this support various, other smaller companies, drug development biotech companies. And we work on various molecules. Some of them, like Mike pointed out, has very high solubility, and it may be difficult to crystallize and isolate. And a lot of them are easy to crystallize but are break dust. So the experience of working with various type of molecules and working on many of them, actually we work on almost over 100 molecules each year through this platform that give us, a lot of experience. So one thing that I want to point out on this slide, we use LC vials to do our the crystallization experiment. It gives us a lot of control in terms of the crystallization conditioning. It also gives us a lot of material for us to do the analysis to assess if they're useful for development. Sorry, I think it's the slide. It has some pictures. It's kind of slow to load. Okay. Sorry about that. It's these pictures, I think, are very large. That's why it takes a long time to load. Again, these are like some specific technology. We isolate our sample on a filtration plate, just like a chemist would do. So we isolate this. We analyze the solid, and this is quite unique in the industry, I think, for those. I don't really have a whole lot of time to get into the details today, but I just want to point out it's quite unique because of time. And this slide shows like various work packages or that you may want to consider at various different development phases of your compound. At discovery stage, a lot of times the real question is, can I crystallize my compound so I can isolate and scale it up? on larger scale? That's typically one of the questions that people will ask. At an early stage, that's really the focus of today, and the discussion is really, we talk about salt and polymorph screening and selection, and find the right salt and right polymorphic form for clinical development. And at a later stage, once your compound goes through the proof of concept, this is a very common strategy for the oncology. People will revisit other indications or different formulations, and that will be another time to really revisit the solid state form. And with that, you can do various studies to support the development objectively. Our platform, again, it's quite flexible. You can do as few experiments. You can do thousands of them, and with the automation, with everything, you can take advantage of that, and it can be done quickly and cost effectively. This actually brings a take-home message, I guess, again, from this is really, do a robust pre-formulation study, and it really lays the foundation for choosing the right delivery technology and formulation strategy for the compound. And first thing is really, know your molecule and know your target product profile. So knowing your molecule is what you have to deal with, and knowing target product profile, even though it may be very preliminary, it's very important to know where you're going, where you're starting, where you're going. So you can do a risk assessment, and you can mitigate various risks by choosing the right solid state form and right formulation strategy for your program. And this will typically get you there faster and more cost effectively, and of course the benefit to the patient. So with that, I think that's the last slide. Thanks for your.
Oh, sorry.
Yeah, thanks very much for your attention.
Just going to say we're about to start our Q&A session now. So if any audience members do have questions, go ahead and type them into the box at the top left-hand corner of your screen and click submit. Now we're going to try to get to as many of these as we have time for. So we've had some great questions coming in throughout the session. Let's go ahead and start with one of these, but do keep them coming in, everybody. This one is, what are the special considerations in peptide drug formulation? PY, do you want to take that one? What are special considerations? Can you repeat the question again? Of course. So the question was, what are the special considerations in peptide drug formulation? Well, the peptide is an interesting type of molecule, actually. It depends on the size. It can be treated as like a small molecule, or if it's a really, larger peptide, it's actually more like a large molecule, like biomolecule-like strategy. So it really depends on the size. Again, no matter, regardless what it might be, you still wanted to do the pre-formulation study, look at the solubility and look at what options that you have and stability and things along those lines. Okay, lovely. Thank you very much. And moving into the next question straight away, how can pre-formulation type information be used in decisions as to whether to redesign a molecule, or when multiple molecules are available, which one to progress?
So I'll go ahead and take that question. I think the question gets back to having multiple properties that impinge on molecular progression. And I think, as has been pointed out, that the preliminary target profile is really important in how you make that decision and use that information. For instance, if you are going into a chemical space or a therapeutic area where it is highly competitive, you obviously put much more stringent criteria on the properties of your molecule because of the competitive nature. If you're going into an area where it's a brand new therapeutic unmet need, then you're willing to take higher risks as you move forward. So by taking that pre-formulation data, you can decide whether those risks make sense to take moving forward.
Lovely. Thank you very much for that answer. Moving into the next question here. This person would like to know when to submit samples for stability indicating method.
I'll give that a step. It's really, you need a stability indicating method to release the material for GLP tox, and certainly for GMP material that's going to be used for the clinical studies. So even before that, you really have to develop a method and validate the method. So to do that test.
Okay, thank you. This next question is, how is the projected dose range considered in conjunction with pre-formulation characterization to assess risk of progression?
As actually was pointed out by both PY and myself in this particular case, the dose becomes critical because you have to combine that with solubility as well that leads into dissolution. And so when you combine those two, I think it becomes important. And I think what we were trying to point out is that you have to combine it, that you can't just take it on its own. That is a very, what might be an acceptable dose for a highly soluble drug is not an acceptable dose for a very poorly soluble drug. And part of the reason for that is as you go to poorly soluble drugs and try to take on formulation strategies, you generally have to incorporate a larger proportion of excipient into those formulations to overcome some of those issues. So I think that the dose always has to play a role, but has to be taken into account with both permeability and solubility, as well as a competitive landscape.
Thank you very much for that. We have a few more moments just for a couple of more questions here. So this one's going to be, there are many CROs with solid-state screening service. What makes Optiform Technologies unique and advantageous?
Yeah, that's actually a good question. Start with, one of my former colleagues at Lilly, basically she said, just about anyone with a powder X-ray diffractometer and LC vials claim they do it, but they don't really do the solid polymer screening. I think the key, again, is really it's a very complex process, and because there are many factors you really have to consider, number one, and the key is really consider them collectively and together. So that's one. The second is really the technology itself, the platform, the way we do it. Most people use microtiter plate to do the screening, and with that, you don't have a whole lot of control on the crystallization experiment, and often you get very limited amount of material and poor quality material for characterization. So with the platform that we have, we started with microtiter plate, just like everyone else, and very quickly we moved back into LC vials, so we can generate high quality material and enough material for detailed characterization to make a good decision. So that's really the platform itself. I think more importantly, it's really your experience. This is really a multidisciplinary area, and you are assessing a lot of different factors in terms of manufacturability, in terms of material property, and also fit into the overall formulation strategy, the overall development strategy. To have a lot of experience with different molecules certainly helps.
Okay, thank you very much for that answer, and thank you to everybody who submitted a question today. Unfortunately, we are all out of time. So that does bring us to the end of the webinar. It leaves me to thank Michael and PY for what was a great presentation, and to thank Catalent Pharma Solutions for sponsoring this session. To the attendees, you will receive an email shortly telling you how you can access the on-demand version of this webinar, or you can access it through our website, which is www.business-review-webinars.com. We do look forward to sharing further webinars with you, so keep an eye out on the website just mentioned. Follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn group as well, Business Review Webinars. Thank you once again, and I hope you all have a lovely day.